Hello, everyone. Welcome to today's live broadcast, Combined Use of Multiple Particle Characterization Technologies to Evaluate Targeted Liposomal Formulations, Implications for General Nanoparticle Development. My name is Sue Pham from LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter Life Sciences is dedicated to empowering discovery and scientific breakthroughs. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. A leader in centrifugation and flow cytometry, Beckman Coulter has long been an innovator in particle characterization and laboratory automation, and its products are used at the forefront of important areas of investigation, including genomics and proteomics. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would like now to introduce today's speaker, Dr. Melvin Klegerman. Dr. Klegerman received a BA in chemistry from the University of Illinois at Chicago in 1968 and a PhD in biochemistry from Loyola University of Chicago in 1984. For more than 30 years, he has conducted biomedical research focusing on immunochemistry, immunoassay development, and conjugation chemistry. For the past 20 years, Dr. Klegerman has been involved in a collaborative effort with cardiovascular researchers under direction of Dr. David D. McPherson to develop targeted immunoliposomes for diagnosis and treatment of cardiovascular diseases, joining the group when they moved to the University of Texas Health Science Center in Houston, where he is now a professor in the Division of Cardiovascular Diseases. As part of this effort, Dr. Klegerman founded two companies to develop the Echogenic liposome technology into clinical products. He is an author of 75 research papers, awardee of 4 patents, and co-editor of a textbook of pharmaceutical biotechnology. I will now turn it over to Dr. Melvin Klegerman for his presentation.
Thank you. Well, good morning, everyone. I'd like to thank you for registering for this webinar. I hope this information will be useful to you. I'd like to start off by giving you an overview of our targeted platform technology, which is echogenic liposomes, which we abbreviate as ELIP. Okay, now first I'd like to give you a sort of review, give you a general review of why molecular targeting. Although I'm sure that most of you have very definite ideas about why you would want to target the applications of interest to you. Of course, there's two major types of targeting: diagnostic and therapeutic, most of the diagnostic applications are imaging related, and among these, there's quite a few having to do with contrast agents, depending upon the type of imaging technology. For instance, if you have ultrasound imaging, then your contrast agent would probably contain some sort of gas. If it's MRI, then it probably contains some gadolinium compound. If it's CT, then it's probably an iodinated compound. And these would be then loaded in the microparticle that you'd be working with. Now, there's a number of diagnostic applications where you have direct conjugation of your ligand, your binding ligand, to the imaging agent, and there's a lot of fluorescent compounds of this sort. These are fluorophores. Recently, there's a particular interest in infrared fluorophores, which have more penetrating capacity. And then, of course, there's radioligands, which is conjugation of radionuclides to ligands. This is the oldest type of imaging technology, actually. But in every case, you can load a nanoparticle with a targeted nanoparticle with the imaging agent. Of course, the major commercial application for nanoparticles is therapeutic. And if you talk about therapeutic targeting, you have to talk about the pharmacologic window, which is the interval between the minimum effective dose of a agent and of a therapeutic agent and the minimum unacceptable toxicity. And I've given some examples here of drugs that are or therapeutics that are in clinical use and the problems involved with a systemic application or systemic dosage, which is still the predominant therapeutic approach. Doxorubicin, anti-cancer compound, cannot be given at more than 550 milligrams per square meter total body surface area because at that point you get 5% incidence of congestive heart failure. And then alteplase, which is recombinant tissue plasminogen activator, which is the only licensed thrombolytic in the United States, is typically given at a dose of 9/10 milligram per kilogram bolus, followed by an infusion, and even at this point you're going to get 4 to 14% incidence of intracerebral hemorrhage, which many clinicians feel is unacceptable to begin with, and has a lot to do with the relatively small market for this therapeutic for myocardial infarction and ischemic stroke. Avastin or bevacizumab is a therapeutic monoclonal antibody that is used for several cancers, and the dose is 10 to 15 milligrams per kilogram every 2 to 3 weeks. And this is nearly a gram of monoclonal antibody at a time, which you're going to get a lot of adverse effects from that alone. But in this case, since this is targeted to VEGF, to inhibit tumor vascularization, you get some pretty horrendous side effects, including gastrointestinal perforation, bleeding, and severe hypertension. So obviously, if you can target these formulations specifically to the pathologic tissue or structures, you could widen that therapeutic window so that you get you get a more effective dose with a minimum of toxicity to other sites or other types of cells than what you really want to target. Okay, now the types of targeting, there's two major types. There's passive and active. Passive targeting is a situation where your nanoparticle, by virtue of its basic properties, will end up where you want it to go. And the major example of this is tumor targeting through EPR effect, and that's enhanced permeability and retention because of the ineffective, leaky tumor vasculature. Particles of a certain size will tend to collect in that vasculature and not leave. And a lot of people are taking advantage of this to structure the size of their particles to enhance this. In atherosclerosis, which is our focus, there are a number of formulations that can end up in areas of turbulent flow in arteries, which is where atherosclerotic plaques or atheroma tend to form. And then active targeting. There's two types of active targeting. There's physical methods, and examples of this is with ultrasound. By holding an ultrasound probe over a particular part of the body, you can fragment microbubbles or gas-containing liposomes that have been injected intravenously. And thereby you release the payload where you want it. However, if you change the ultrasound parameters, you can to a lower level that will not destroy these vesicles. You can use the radiation pressure of the ultrasound to force these vesicles to the vessel wall, where they can then be more effective. For various reasons, you can have a slow release or a slower release of the payload. There's a lot of work recently having to do with magnetic attraction. So again, the same principle. You can hold a magnet over a particular part of the body, and then you can formulate your nanoparticles with a paramagnetic compound. Then, of course, the other aspect of active targeting is chemical methods, and this is mainly covalent conjugation of your targeting ligand to your nanoparticle. And so, although the actual association of the ligand and the target molecule is non-covalent, and so therefore, a major aspect of this is, and this is what we'll get into, is the affinity of binding. And it needs to be high enough to be clinically useful. And it depends on the application. So for cancer applications, you have a natural advantage in that you're dealing with a resting situation for the most part. And you can get away with lower affinities than you can with, for instance, cardiovascular applications where you're dealing with arterial flow rates, which can be fairly high. And so therefore you have to have an affinity which will counteract or overcome the shear stress, the lateral shear stress involved. And this generally means that it needs to be sub-micromolar. This is still an issue that is not, I think, widely appreciated. A lot of people are doing more of semi-quantitative or almost qualitative approaches to demonstrating targeting. And this really, for clinical applications, this really needs to be more quantitative, more specific. So, and then we'll get into the kind of evaluation that we're doing for that, and maybe you can get an idea of what the value of that might be. And then, of course, you don't stop with affinity, because affinity that you measure, okay, so as I say, for shear stress, it's sub-micromolar. In most cases, you want it to be 1 to 10 nanomolar. If you can get it higher, that's good, but it's not easy to get higher than that. But you're not just, this is when you're talking about affinity, you're talking about monomolecular intrinsic affinity between the ligand and the target. But really what you're talking about is an avidity. This is also an issue that is not well understood. Avidity is the actual aggregate affinity of a number of ligands that are actually binding to the target molecules on a surface. And of course, this is the product of the monomolecular affinity and the number of ligands that act on a nanoparticle that are actually binding to the target on a surface. And then beyond that, we have done thermodynamic analysis, which is also not widely used, but this gives a lot of information about the environment of binding and what's the best ligand to use in a particular environment. For instance, if you have an entropic association, which is going to be hydrophobic, then you're going to get the maximum binding strength in an aqueous environment. Whereas if your binding is going to occur in a relatively apolar environment, then you'd be better off with a highly enthalpic interaction, which would involve ionic interactions. So you'd want charges and so on. And then in terms of the actual linkage, the covalent linkage of the ligand to the nanoparticle stability issues are primary. So, and we're talking about storage and then in vivo stability. And this can be complex because, like for instance, in cancer applications, a lot of times you want the bond to be stable in the circulation, but then once it's incorporated into a cell, you might want the bond to be broken so that to liberate the actual therapeutic agent, so that it can have maximal activity within the cancer cell. And there are a number of formulations that have been made so that the bonds will break inside a lysosome, for instance, at a lower pH. So these are some considerations for targeting as an introduction. And then now our applications, as I say, involve atherosclerosis. Now, a modern understanding of atherosclerosis is that it's an inflammatory process. And therefore, you're going to end up targeting inflammatory markers, which are mainly integrins and adhesion molecules. And I'll get into that, examples of that, of what we target. And it's not just in the inside of the arterial wall. So I'll make a little arrow here. The inside of the arterial wall, which is right here, next to the blood flow, but also these markers are on the outside, and then there is a capillary network that goes through the wall, known as the vesivosorum. So you would want your nanoparticles to be able to go through this network and be able to impact the atheroma, or the atherosclerotic plaque. I should use, okay, so now, now our liposome, as I said, we are working with intrinsically echogenic liposomes. And what that means is that by virtue of the production method of these vesicles, the air becomes trapped in both the lipid bilayer and in the vesicular lumen. And therefore that reflects ultrasound. The gas is usually used because it's highly reflective of ultrasound, and that makes it a contrast agent. Now, the advantage of liposomes is that they can easily be conjugated to ligands like antibodies, and through the ethanolamine head groups usually of the phospholipids, and then so they're very convenient that way. And then here's some proof of principle involving our ELIP. That's first of all the echogenicity in vitro. There's two types of ultrasound imaging. Intravascular, intravascular ultrasound, or IVUS. This is where you have a transducer, an ultrasound transducer that is incorporated into the tip of a catheter that can be inserted into the arteries and threaded to whatever arteries, such as coronary arteries or the carotid artery that you're interested in visualizing. Now, in vitro, you could simply take a suspension of these liposomes and put them into a vial. And then using one of these catheters, you can show the reflectivity of the agent. And this can be quantitated because then we could just do video densitometric methods to get a mean grayscale value for the average area of this. And this just indicates that our ELIP, in terms of acoustic reflectivity, are actually superior to commercially available contrast agents such as Optison or some of the other commercial, Definity is another one. So then, And then the second type of ultrasound imaging is, in this case, transvascular ultrasound, but this is just one type of general ultrasound, of conventional ultrasound, in which the probe is held outside of the body. And in this case, a well has been punched in a gel standoff pad, and then you hold the probe lateral to that, and then you can see the reflectivity. And so then in terms of targeting, this is a scanning electron micrograph of fibrin-targeted ELIP binding to a fibrin network on a fibrin-coated slide. But now in vivo, we are working with a Yucatan mini swine atherosclerosis model, which is very similar to the human situation. And so in this case, we induced a fibrin thrombus or clot in the femoral artery of one of these small pigs. And then immediately after injection of fibrin-targeted ELIP, you could see how it just lights up, and you get intense reflectivity. And then here's a case for the transvascular ultrasound, where the probe is held on the neck of the animal, and this is the carotid artery. And in this case, an atheroma has been induced in that artery. And then in this case, we're working with ICAM1, which is an adhesion molecule, an inflammatory marker. So it's ICAM1 targeted ELIP, and after injection, they bind to the atheroma site. So it's very easy to see. So this is something, a platform technology, and here we're using 3D IVUS to actually show the atheroma on the surface of the vessel of an artery. So that this is a platform technology we've actually been working on for about 25 years now, and these are just the diagnostic applications. I won't go into therapeutic applications, but the point is give you an idea of what the targeting that we're doing, and the kind of nanoparticle formulation that we're working with. Now, okay, so now I'll get into the evaluation methods that we use. Obviously, for this, first we look at echogenicity. So this is a particular contrast agent. And the echogenicity is important for therapeutic use also, because by applying a particular ultrasound to these, once they're at the target, you can release the payload. So it's a controlled release formulation as well. Okay, like I say, the echogenicity is done with in vitro IVUS. And then we get into the particle characterization. So we use the Beckman-Coulter Multisizer 4 to evaluate our liposome population that's larger than 0.4 micron equivalent spherical diameter. And then and the important thing about the Multisizer is that it counts the particles. This is very important in terms of the kind of parameters we're looking at. And then there's the particle size distribution. And of course, I'll show you these. We use the median diameter rather than the mean diameter of a number distribution, and I'll get into why we do that. And then, and then we use dynamic light scattering. In this case, Melvin Zeta sizer, to look at all the liposomes that are smaller than 400 nanometers. However, these DLS instruments do not count particles. You do not get the enumeration. However, what you do get is you get the fraction of the total distribution, like in a number distribution for each peak, which is very important again, and I'll show you how we use that. And then the actual range of the Zeta sizer is about 4 nanometers to 10 microns. This is also important because there's an overlap in the range between the Multisizer and the Zeta sizer, which is I'll show you how we use that as well. Okay, and then conjugation efficiency. So if you're doing targeting, you need to know, and like I say, a lot of people do a qualitative or a semi-quantitative approach to these things, but it's very important to know exactly what your conjugation efficiency is, how much of your ligand is actually conjugated to your nanoparticle. And then, so we have had to develop our own specific quantitative immunoblot assay because phospholipids, which are used in liposomes and many other lipid formulations, interfere with all the standard protein assays. So we have developed an assay where we're actually measuring the protein on the surface, conjugated to the surface of the liposome. And then with that, and then we get a result that's in micrograms. In this case, we're mainly talking about antibodies. So we're talking about micrograms of IgG per milligram of lipid. That is our primary conjugation efficiency index. However, we go from there to because of the particle characterization techniques that we're using, we can then go from there to determine the number of IgG molecules conjugated per liposome. And then we have to do this for both size populations. And the reason we work with a more complex size distribution than a lot of people do because of the ultrasound aspects of it. Because we actually have particles that go from less than 100 nanometers to more several microns in size, we can take advantage of many different frequencies of ultrasound to be able to characterize them and to monitor them. Now, then we get into targeting efficiency. And this really gives you a lot of advantages if you can determine actual quantitative targeting efficiency, which means the liposomes that are actually binding to a mobilized antigen. We use a quantitative ELISA method, which actually will measure the antibody affinity, starting with the dissociation constant, which is easily converted to the association constant, which is important for avidity measurements. And then we also determine a direct, direct targeting efficiency, which is the ratio of the ELISA measured amount of specific antibody relative to the total amount of IgG measured by the Immunoblot Assay. And from this, we get 3 different indices, targeting indices. There's the specific avidity, which is the total number of antibodies conjugated per liposome times the association constant. So then you get an overall avidity. However, that's not specific enough. So we go to a specific targeting efficiency, which is based on this direct targeting efficiency, and this gives us the number of antibody molecules that are actually binding to target molecules in the ELISA. And the ELISA actually is very similar to the physiologic situation in the sense that we are measuring the antibody that is conjugated to the whole liposome, and then that is actually binding to the antigen molecules on a surface. So that is actually fairly more close to the physiologic situation. And then again, we take this number of antibodies and multiply it by the association constant, we get what we call a functional avidity, which is the actual affinity of the ligand on the liposome that's binding to antigen on a surface. And we find that's the most informative targeting index of all. Okay, so here's some examples of our results. Echogenicity, like we're using mean grayscale value here, and the untargeted ELIP will give you a mean grayscale value will maximal, will top out at about 200 out of a maximum of 255. And we've gotten our targeting procedure to the point of where the echogenicity of the targeted ELIP are almost as good as the untargeted. So they're very echogenic. And we use what we call an index, which is E50, which is the echogenicity and mean grayscale value for 50 micrograms per ml suspension of the liposomes. Now here is the Multisizer report. Unfortunately, you can't see the numbers too well, but here's the distribution. And what this is actually is the junction between two populations. This is actually a normal distribution of the larger particles, and you could see that the mean of this is around 650 nanometers. And then you start the much larger population of the small liposomes. And so we have found that the median, can't really see it here, but we have found that the median is the closest to the actual mean of that larger distribution, because you're looking at a combination of the two populations. And so in this case, in this report, it's 645 nanometers. And then here's the Zeta sizer, and you can see that there is definitely, it's bimodal. There are two distinct peaks here. This is a volume distribution. So this will show both with actually the emphasis on the larger population. But if you look at a number distribution, the smaller liposomes dominate, and they are less than 100 nanometers in size, whereas you can just barely see the larger ones. However, the size corresponds very well to the Zeta to the Multisizer. It's about 650 nanometers, and then this shows, and again, it didn't come out too clearly, but this shows that the smaller liposomes represent 98.9% of the total number of liposomes, and using that number, we can then calculate from the Multisizer results. We can calculate the number of the smaller particles that are below the range of the Multisizer. And then here is just to show you what the Immunoblot assay looks like. These are immunoblots of liposomes after purification, and we do densitometry of the blot, and we find the semi-log plot is linear, and our effective range is from 0.3 to 4 micrograms of IgG per ml. And so using that, we can calculate the conjugation efficiency in molecules per liposome for both populations. So what we do, as I said, is we take the number of liposomes smaller than 400 nanometers based on the fraction in the Zeta sizer and the percent number distribution and the number of liposomes counted by the Multisizer that are greater than 400 nanometers. And then from that, with the particle sizes that are determined by each method. So the small, in this case, the small liposomes average about 71 nanometers in diameter, and in the larger population average about 650 nanometers. And based on that, we can calculate the total surface area of the liposomes and determine the fraction of that total for each population. And then it makes it very easy. Well, then we can convert the Immunoblot result to molecules of IgG per square meter, and then it's very simple to just convert that to number of IgG molecules per liposome average. So it's the average number of molecules of IgG per liposome for each population. Now, the reason we do this, we're really interested in mainly in the larger population, because in terms of echogenicity and drug loading, that is really the population that is most useful. But we need to know how much of the antibody or the ligand is conjugated to the smaller liposomes so that we get an accurate conjugation efficiency for the population of interest. So, for instance, we calculate that the smaller liposomes have normally 5 to 20 molecules of ligand, of IgG, bound per liposome. And it's really, for our applications, that's very marginal. Whereas, and I'll show you, whereas for the larger, the population, you're talking about hundreds to thousands of molecules per liposome. And then here is the affinity calculation from the ELISA. It takes advantage of the fact that the ELISA relation is hyperbolic. And you simply use an equation for hyperbola, and with the B parameter being the X value at 1/2 maximal Y is the dissociation constant. That's based on the assumption that the optical density of the colored product of the enzymic reaction is directly proportional to the binding, and we have validated this with other methods, with a radioimmunoassay technique, which is in many ways the gold standard for affinity measurements. And here is, unfortunately, it's a little hard to see, but here is a composite of all of our indices for conjugation and for targeting for a number of targeted ELIP batches. And so, we have a number of targets: fibrinogen, fibrin, ICAM1, VCAM1, which is another inflammatory marker, and then alpha V beta 3 integrin and VEGF. And then we using a number of different antibodies. But here, the conjugation efficiency, micrograms of antibody per milligram of lipid, we have found that 10 and higher is optimal. And so there's a number of these formulations that that achieve that. Number of antibodies, and these are all greater than 400 nanometer in size. So that's the population we've got here. So we found that 1,000 antibody molecules per liposome in that population is optimal. So then the average size of the liposomes tends to be much more consistent. We find they usually fall between 500 and 800 nanometers in size in diameter. And then the number of liposomes per milligram of lipid, 3 times 10 to the 9th and above, is optimal. And then we get into the targeting parameters. So this is the specific avidity, and using a physiologic flow simulator, we have shown that 2 times 10 to the 12th liters per mole per liposome is optimal. And this is not easy to achieve. However, anything approaching that value will give you a very useful formulation that gives you good binding and functional utility. Then we get into specific targeting efficiency, and based on the fact that the maximum practical surface area of a liposome that can bind to a surface is about 40%, then that would indicate that at least 400 molecules would be optimal. Again, that's not easy to achieve, but most of our formulations approach that value. And and this is actually not specific, this is functional avidity based on the same principle. 10 to the 11th liters per mole per liposome would probably be optimal. And it's the same story. I mean, you approach it often, but there aren't too many formulations where you can actually get that high. Again, it depends on your application, depends on your formulation. So, and you have to more or less work out what is optimal for you. Now, the final determinant of this is an animal model, in vivo, and in our case, we have a lot of data having to do with the enhancement of imaging in this atherosclerosis model that we're using, and so that should be correlated with that. But these are in vitro characteristics that can be used to optimize and characterize whatever nanoparticulate formulation you're working with. So that's all I have to say here. I hope this is of some interest, and that you can see where you might be able to apply at least some of these things to your specific application. So at this point, I guess I can take some questions, or if anybody has any questions.
Well, thank you, Dr. Klegerman, for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. We already have a few. And our first question, one viewer says, great technique. What is the efficacy or selectivity of ELIP liposomes targeting when they are conjugated to targeted antibodies?
Well, yes, that's this selectivity, or the ability to. We have shown, a number of publications that you get in terms of the diagnostic or the targeting aspect, you get a significant increase in the highlighting of the Atheroma, relative, and then we use a number of controls. So we use liposomes, typically liposomes that are conjugated to non-specific IgG as controls. So these are fully echogenic, but they're not specifically targeted. However, they do have protein on them. So, and then you find relative to that, for instance, for ICAM1 targeting, we often find a 40% enhancement of echogenicity or highlighting. And again, what we do with this is you take the ultrasound sonograms, such as the IVUS, and then convert them, by video densitometric methods to mean grayscale values. And we have an ultrasound engineer who's very, has developed a lot of these methods, and therefore you can take ROIs, regions of interest, and then do the image analysis on them. Of course, a lot of times you can see this visually, but you would ultimately have to use some sort of hardware and software in order to be able to instantaneously determine highlighting in clinically. But that's for instance. Now, in terms of we've also done a lot of work therapeutically to demonstrate that the targeting that the targeted formulation of a drug or agent is significantly greater than the control or the drug alone, and which is what a lot of people do, of course. Ultimately, that you have to see what is your enhancement factor for your formulation. So I hope that answers the question.
Great. Our next question, one viewer asks, what is your preferred technique for counting nanoparticles?
Well, the Multisizer is the best out there. Now, I didn't talk about what people typically use to enumerate their nanoparticles. A lot of times they use electron microscopy, which is rather laborious, and especially when you have to count 300 or more particles to get a meaningful or a useful distribution and a number. So what people, I think, usually do is they try to relate using a gold standard method like that, then they try to relate the number of particles to mass or some property. For instance, if your particles for some reason are fluorescent, or you can determine the quantitative fluorescence of them and then relate that back to number of particles. You'd have to have some consistent size, so that your method, your production method, will pretty much consistently guarantee you a certain size distribution, which you can also do with a DLS instrument. You can then determine the size of your particles, and then use something that's based on mass or that's based on something that can be quantitated and work that back. However, the Multisizer is really cannot be matched in terms of the ability to immediately connect distribution data with size distribution data with number. And of course, we have, for instance, we have collaborators who actually go into the individual bin numbers. They will take the data, and they will determine how many particles are in each in each narrow interval. So I mean, you can. That's the advantage of that. You can deconstruct the data in a way. The instrument will allow you to do this, that you can look at your data any way you want in terms of size and number. And I'm not aware of anything else that can do that.
Great for its flexibility. Subagini asks, in vivo or in vitro, what is the level of non-specific binding and fusion of antibody-conjugated liposomes to biological membranes that do not present with the antigen?
Yes, that's and of course it depends on the tissue. It depends on the situation. Like for instance, you have some tissues that tend to be sticky. We have found, for instance, that porcine. In certain situations, it tends to be higher non-specific binding than others. But in every case, your specific signal has to be significantly higher than the non-specific. So that in terms of echogenicity, for instance, in the kind of atheroma work or the atherosclerosis work we're doing, you can get a non-specific binding of, let's say, 15%. Well, it depends, of course. If you're working with mean grayscale values, in many cases, you might get something like 15 for your non-specific, and then you will and then you look at your specific binding, and this can be 80 or something of that sort. But we tend to, when you're dealing with different animals, from one animal to another, percentage is the most informative. So what you're looking at is the percent enhancement relative to a control. And and then you talk about, so your baseline tissue without any liposomes. It varies. It varies as far as, but like I say, for porcine arteries that may or may not be atherosclerotic, you'd get let's say a baseline of around 15 using that scale, or 10, 15, 20, and then your non-specific with your liposomes might be a little higher, maybe 30, and then your specific are up to maybe 80, 60 to 80. I mean, just giving you some numbers that we have seen. Yeah, there is definitely a non-specific echogenicity to tissues of all kinds, and you have to measure that, and you have to calculate what it is above that, and it has to be significant and useful. It has to be useful. So there must be a way of translating that into clinical use. So I hope that might have answered the question.
That was great. We have another question that asks, are there any other characterization equipment apart from BC Multisizer and Melvin Zeta Sizer?
Oh, there's a number of dynamic light scattering instruments that are out there. We used to use a NICOMP years ago, and I have to admit, I'm not sure. In the Zeta size, Melvin makes a number of, and I don't want to be advertising for any particular, because there are quite a few of these instruments. And then there's not just dynamic light scattering. There are other small particle characterization techniques that are used, and a number of them use lasers. And of course, there's the flow cytometers. And a lot of people are using flow cytometers now. Of course, Beckman makes a number of these. A number of people who are working with flow cytometers to do quantitative particle enumeration and sizing. Of course, it needs to be fluoride. You have to have a fluorophore, targeted fluorophore, to work with these things. But there a lot of people are using it, and so that's another particle sizing technology that's being used more and more. There are, and so of course the flow cytometers, such as those made by Beckman, use the Coulter principle. So that's why you can enumerate the particles. So that's, of course, you're also doing it by fluorescence and fluorescence sorting. But it's, and then there's the cell sorters that actually will retrieve your particles of interest, and cells. So yeah, there are quite a few different sizing technologies, and Coulter has a whole range of counters and sizers, and there are those that are designed for working with larger particles, such as cells, and so on. So we use the Multisizer because, first of all, it will go down to the smallest size possible with the Coulter principle, which is about 400 nanometers. Of course, the problem with that is you're using an aperture tube, and the smaller you get on an aperture, the more likely it is to clog. So there are some technical issues, but it's easy to get proficient at that dealing with. And now the Multisizer 4 is terrific. Most of this has been automated, so that if your aperture gets clogged, it'll just unclog it automatically for you. But you have to be careful. You have to work with very clean preparations and clean solutions to get around that. So, but this is, but in terms of, like I say, being able to enumerate and size at the same time to great detail, the Multisizer is probably the best instrument we've worked with. But there are many different sizing, and like I say, you want to look at the small populations of nanoparticles that will give you size distributions with proportions for each peak. It's not just the dynamic light scattering instruments. There are a number of other instruments that will do the same thing as well.
Great.
I just want to say, as far as where you would get an overview of that or a review, it's, you could search on the web. I mean, there's really a lot of information out there.
Great. Our next viewer asked if you could explain how you measure protein concentration on the surface of liposomes. And in addition to that, they ask how we could separate the complexes containing liposomes and proteins from the liposomes alone.
Okay, that's a very good question. What we do, of course, this Immunoblot Assay. So, okay, well, first I should talk about how we purify the liposomes. Now, well, we've used a number of different methods for that. The first method we used was gel chromatography. So you could use a Cepheros column, and then you put your liposomes through the column, and then you can separate by size. So liposomes, of course, are very large. They're certainly more than 1 million as far as equivalent to molecular weight. And then it's very easy to separate free antibody from the liposomes that way. The problem with that is that you get a large dilution. So then you end up with a relatively dilute suspension of liposomes. Now, there are ways of concentrating liposomes depending upon your application. We can't do that because ours are echogenic, and there's a whole lot of things that will destroy the echogenicity, any kind of centrifugation, any change in pressure, so on. So we went then to dialysis, and there are large pore-sized dialysis membranes, but they tend to be Teflon-like substances that are very hard to work with, and the supplier has stopped using, making the kind of dialysis units that were very useful to us. This is but that worked rather well while we were doing it. But now what we're using is a method, and that involves a small Cepheros column, Cepheros CL4B, which has been preconditioned or preloaded with plain liposomes, with untargeted liposomes. And this enables, the liposomes to come through intact relatively quickly, but you have to calibrate it very carefully so that you exclude unconjugated antibodies. So these are some of the methods that we use. But now, there's a whole lot of other methods that are possible for people who don't have to worry about things like echogenicity, including centrifugal methods. So there are a number of ways to purify liposomes. And then, okay, so once you purify them, then our Immunoblot assay, you spot the liposomes on a membrane, and then you can you then develop it, and then you get the purple color. We work with alkaline phosphatase. You can also work with horseradish peroxidase substrates. As far as whatever your Immunoblot method might be, it's the same basically as Western blot. And then you just used densitometry to quantitate the color intensity. Now, in terms of standardizing these, this is the trick. In terms of standardizing this assay, and we have a, if you can access the slides, then you can see what the reference is. We had to use radio tracer methods. So in other words, we did a conjugation of the liposomes with a radio-labeled IgG. And then we used that to determine, to come up with a primary standard of exactly how much IgG was on the liposomes. And then from there you can make secondary standards, so that once you have, and then we use a composite standard curve, which is what I showed, which is a curve that is reproducible. You have to use the same methods every time, and then you can you have an equation, and therefore you use this to quantitate. But using these methods, you then make secondary standards which you can store, and then just use those. But periodically, you might want to re-standardize with another radio-labeled preparation. So that's how we did that.
Right. Our next question, one viewer asked, have you ever used spectroscopic quantitative analysis?
Spectroscopic. I'd have to say no, we really haven't. I mean, we've, I think I need to know more specifically what, I mean, we use spectroscopic methods such as GC mass spec, but we haven't used it for this particular application. So I guess I'd have to say we really haven't done that.
Okay. Well, unfortunately, we have more questions than we do have time for.
Oh.
But we'll be sure to get to the questions later on. At this point, I'd like to pass control back to Dr. Melvin and ask if you have any final comments.
Well, there were a couple of things that I did not mention, but I might as well now. One is that I'm not sure how many people here. A lot of people are actually, I mean, even in the liposome field, you're going to be working with a monomodal distribution, and in many of these new applications, these nano medical applications, you're working with particles that are well below 400 nanometers in size. So in a case like that, people might think that the multisizer would be of no value to them. However, in many cases, you might also be already using a multisizer for other applications, so you have one on hand. Now, there is a little trick you can do with this. You can, so like I say, you might have a very small population of very small particles, including liposomes. A lot of liposomes now are extruded so that you end up with a single sharp distribution, and it might be below 100 nanometers in size. But what you can do, so you're going to be using something like DLS instrumentation to to evaluate these, to characterize them, but you can't enumerate them. And like I say, there are other methods people do use. But what you can do is dope that suspension with, like, let's say, 2-micron latex beads. And that you can enumerate. So you take that, and that you can enumerate in the multisizer, and then go back and get the distribution for both peaks in your DLS instrument, let's say, and get the fractions, number fractions, and then use the same method to determine the number of your nanoparticles, and then you can validate this with something like electron microscopy, one of these gold standard methods. And if it works, then that you could still use the multisizer and it'd be of great value. And then the other thing I'd like to say is that we are still working on refining our targeting parameters, so that what we really want to get to is determining the force of binding. And now, since we have avidities, working avidities, these can be converted to free energy, and that can be converted to force. And then the idea, ultimately, what we want to do is to be able to get actual binding force that can be tested with atomic force microscopy, for instance, and have parameters that are, and a lot of people are working on those kind of considerations. And then we can start looking at lateral shear stresses to optimize these targeted formulations. So I hope that's, the way, sort of the way of the future, that this using quantitative methods because, the problem is that with all these targeted formulations, they're not getting into the clinic. They're usually breaking down, when it comes to even late stage preclinical studies, but certainly for clinical trials. So the more you can understand quantitatively about your formulation, the better chance you have of actually ending up with something in the clinic. So I guess that's all I have to say on this.
That was very informative. I would like, once again, to thank Dr. Melvin Klegerman for his presentation. Today's webcast will be available for on-demand viewing through July 2016. You will receive an email from LabRoots alerting when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.
Thank you. Goodbye.